processes occur in certain parts of the cell. We know that eukaryotic cell has some organelles and components, the main components being the cell membrane, the nucleus, and the cytoplasm. Here's a picture of the cell structures. You see here the membrane is kind of the barrier for the cell. It ensures that the cell keeps its boundaries, and this membrane allows for nutrients to enter and waste products that exit the cell. We have the nucleus. We also have the mitochondria, and those are some. Oh, and the cytosol. Excuse me. The cytosol is the aqueous or liquid portion within the cell. Those are the main areas that we're going to talk about in terms of where metabolic processes occur. Here are some other just definitions or structures and their functions. Many of these, this should be a review. You guys probably learned about this in biology, but for purposes of what we're going to be doing in this block, we're going to be looking at the different areas, particularly the cytosol or the mitochondria, where many of the reactions will be occurring. So in the last lecture of block 1, enzyme lecture, we talked a lot about coenzymes and cofactors. Coenzymes and cofactors are very important, and they work with enzymes in order to carry out all of these metabolic processes. We know that coenzymes are vitamin derivatives. For example, we talked about nicotinamide adenine dinucleotide, which is a niacin or vitamin B3 derivative. We're going to talk a lot about how these cofactors regulate or are involved in the regulation of the metabolic processes, and exactly what they do, because they have an important function in terms of energy production. So also from the last block, we talked about the different types of enzymatic reactions. We have the oxidation reduction reactions, or redox for short. And so what happens is the coenzymes will either be oxidized or reduced when they are involved in carrying out a reaction with an enzyme. And so the characteristics of oxidation are shown here. Oxidation would be the loss of electrons and or hydrogen ions, the gain of oxygen, and the release of energy. So an oxidation reaction can have all or one of these components, and that's enough to make it an oxidation reaction. For reduction, you see that there is a gain of electrons and or hydrogen ions, a loss of oxygen or input of energy, and so a reduction reaction contains one or all of these in order for it to be a reduction reaction. And so at the bottom, here's some little mnemonics to help you know what's going on in each oxidation and reduction reaction. So for oxidation, you gain an oxygen and you lose hydrogen or electrons, OHE. For reduction, you gain hydrogens or electrons, and it's a reduction reaction and you lose oxygen. Any questions so far? So you do not need to know these structures. This is the structure of nicotinamide adenine dinucleotide. The take-home message is that when it's NAD+, that is when it's in its oxidized form, and when it's reduced, it's NADH plus H+. Why is it reduced? Because it gained hydrogen and electrons. So it gains hydrogen and electrons, right? Does that make sense? So this is the reduced form of the coenzyme, NADH plus H plus. I might just say NADH for short, to shorten it. And so how this enzyme or this coenzyme carries out its reaction with actual enzyme, we saw this. This was an example given in the last lecture. So ethanol or alcohol in the presence of the enzyme alcohol dehydrogenase. Alcohol is oxidized, reducing NADH to NADH plus H plus. And so that converts alcohol or ethanol into ethanol, which is a C double bond O. Again, the take-home message is that NADH plus H plus is the reduced form of the coenzyme. So this is very important, and we're going to talk about why as we progress in this lecture. NADP, or nicotinamide adenine dinucleotide phosphate. Same structure as NAD, except for there's a phosphate group here. You don't need to memorize the structure. When it is reduced to NADPH. So NADP is the oxidized form, NADPH is the reduced form. This coenzyme is involved in anabolic reactions. We're going to talk about some of those reactions as we continue in this lecture. FAD is another coenzyme that's derivative of what vitamin? Anyone?
Flavin.
B2, vitamin B2. So this FAD is the oxidized form. It is reduced to FADH2. Oxidized form, reduced form. And this reaction or this coenzyme is involved in the Krebs cycle. Does anybody know what the Krebs cycle is? Another name for it is what?
Citric acid.
Citric acid cycle. Yeah, I heard somebody say unfortunately, and I saw faces. Trust me, I get it. I had to learn this over and over and over and over, including drawing the structures, which you guys do not have to worry about. Thank God. Again, I don't want to pass that misery or bane of existence on to other people, right? So this coenzyme, FAD, is involved in the Krebs or TCA citric acid cycle. We're going to talk about this in the third lecture of this block. And the substrate is succinate. Succinate, in the presence of succinate dehydrogenase, will reduce FAD to FADH2, producing fumarate. FADH2, again, here's the take-home message. NADH plus H plus and FADH2 are very important because they transport electrons to the electron transport chain for ATP production. We're going to talk about that in the fourth lecture of this block. But that's kind of like the take-home message of why these coenzymes, part of the reason why these coenzymes are so important. They're energy reservoirs. So we have another coenzyme, derivative of vitamin B5, pantothenic acid, and this coenzyme A. We know that this thiol group is the reactive group of this molecule. You don't need to know the structure. It's a big structure. But the take-home message for this is that anybody know what acetyl-CoA is? Or where it's found? Say again? In the mitochondria. What is it? Do you know what it does? If you don't, that's okay. We'll talk about it. So,
And the precursor to the citric acid cycle.
acetyl-CoA is a precursor to the citric acid cycle. You're correct too. That occurs there. So what happens after the pathway we're going to talk about today? Glycolysis. Glycolysis is the oxidation of glucose. Glucose is broken down into two pyruvate molecules, and what happens is that pyruvate molecule, if oxygen is present, is converted to acetyl-CoA, and that is the intermediate step transitioning us from the glycolytic cycle to the TCA cycle. We're going to talk about all of this. So, this is a portion of the metabolic map, and you guys are going to know all of this by the time we finish with biochemistry. Isn't that cool? No? You guys will know the important points, the important features of this metabolic map. This is just a piece of some of the pathways we're going to talk about in biochemistry. But we're going to go over these, and I want you guys to focus on the main takeaways that are on the slides, some of them in boxes. You don't need to know the structures of any of this stuff, just need to know the purpose of these pathways and the main products. And we're going to talk about all of these, but we're going to start with glycolysis. Here's a summary of all of the metabolic pathways that we've seen in the previous slide. The ones in green are the pathways we're going to talk about in this lecture. And so I'm going to go over all of this information in green as we progress in the slides. So first pathway, glycolysis. Glycolysis is the oxidation of glucose into two pyruvate molecules, and it produces a small amount of ATP, and it produces NADH, which NADH also is an energy reservoir because it is transported into the mitochondria, into the electron transport chain, and is involved in the production of more ATP molecules. So, what's the purpose of glycolysis?
Energy.
One person, just one person tell me what the purpose of glycolysis is. Say it loud.
To break down glucose.
Break down glucose and what? That's
So
correct.
then you can provide more other molecules for the next cycle, the TCA cycle or the pentose pathway. Something.
Correct. So glycolysis breaks down glucose, producing ATP and NADH, which also will eventually produce ATP. So here's the takeaway from glycolysis. We have this sugar or glucose molecule here. It's broken down into 6 carbon sugar, broken down into 2 3-carbon sugars. And again, if this is done in the presence of oxygen or under aerobic conditions, then the pyruvate will be converted into acetyl-CoA and enter the TCA cycle, the citric acid cycle. Any questions so far? Okay. So, there are 3 irreversible steps in glycolysis, and that is due to the Gibbs-free energy model of thermodynamics. Basically, what this model is saying is that when the products from a reaction are much lower in energy than the substrates, then you have a total energy that is a net energy that is negative. And so all that means is that the reaction is irreversible. So if you have an energy, a total net energy where it's 0 equal to 0 or it's positive, that means the reaction is reversible. But in glycolysis, there are 3 reactions where the total net energy of the product is less than the substrate, so the net is negative. And so that means that these reactions are irreversible. There are 3 of them. The first, the third, and the 10th reactions of glycolysis are irreversible. Hexokinase is the first enzyme in glycolysis that converts glucose to glucose 6 phosphate. In liver cells and the pancreatic cells, hexokinase is called glucokinase. I'm going to say that again. In liver cells or pancreatic cells, hexokinase is called glucokinase. This enzyme is inhibited by high levels of glucose 6 phosphate. Step 3, the enzyme is phosphofructokinase. It's an allosteric enzyme. What does that mean? One person tell me what allosteric enzyme means. Yes. Correct. So the substrate binds to another site on the enzyme and it alters the shape of the active site. So PFK or phosphofructokinase is an allosteric enzyme that's inhibited by high levels of ATP and increased by high levels of ADP or AMP. Pyruvate kinase is the final step in glycolysis. It is also an allosteric enzyme, and it is inhibited by high levels of ATP and acetylcholine. Here are the important components of the glycolytic pathway that I would recommend you guys knowing. The steps in red are the steps that I need you to know. Now, you need to know the irreversible steps, of course, that we just went over. However, step 4 is here just to show you where the 6 carbon sugar is being broken down into 2 3-carbon sugars. Because remember, one glucose molecule produces 2 pyruvate. Does that make sense? So we're going to go over this. So hexokinase, the first enzyme, converts glucose to glucose 6-phosphate. Then in the third step, fructose 6-phosphate is converted to fructose 1,6-bisphosphate by phosphofructokinase. Remember, this is an allosteric enzyme. This product is broken into 2 3-carbon molecules by aldolase, producing DHAP or dihydroxyacetone phosphate, or G3P, glyceraldehyde 3-phosphate. And then finally, step 9 of glycolysis, phosphoenolpyruvate, or PEP, is converted to pyruvate by pyruvate kinase. This is also an allosteric enzyme. Any questions on what I want you guys to know about this slide? So, just, yes?
Do we need to know if each of those enzymes is causing a reduction or an oxidation of a certain thing?
No. So, I'm probably not going to ask about this substrate, right? I shouldn't, right? Anybody?
No, of course not.
Of course not. Why not?
Because you said
Talk about it.
Because I didn't talk about it, and it's not one of the 4 steps, right? Okay. So remember the first lecture, you guys remember everything I said. The first lecture in biochemistry, we talked about carbohydrate metabolism in the GI tract. We talked about transporters, remember the transporters that bring the monosaccharides into the body. We talked about the blood transporters, right? And the SGLT transporter. I remember that, right? Yeah, I like that. Yeah, of course. So, what I want you guys to know from this slide, this is actually drawn by my predecessor. This takes a lot. This is a textbook quality slide. So, I'm very impressed that he drew this manually. But what happens is that glucose is in green. We're just going to focus on the transport, the glucose transporters. So in the small intestine, the enterocyte cells will have the glucose transporters come to the surface, and they will bring glucose into those cells. Glucose will enter the bloodstream, and then glucose will then be brought into an actual tissue, and within the tissue into a particular cell. And what happens is when glycolysis starts, the first reaction does what in glycolysis? What's the product of the first reaction of glycolysis? One person? Glucose 6-phosphate, correct. So when that phosphate is added to the glucose molecule, it traps the glucose into the cell. So that means that glucose is stuck in the cell, and that means it's probably going to continue on in the glycolytic pathway. So again, when carbon number 6 is phosphorylated, once glucose enters the cell, it is trapped in the cell. Any questions? So what carbon number is phosphorylated to trap glucose in the cell? 6. Okay, so if you guys see this again, if anybody misses it, if it's on the test, might be, might not be, there's going to be some problems. Okay, any questions about that? Yes.
So, not to beat a dead horse, but the biggest takeaway here is what's underlined.
That's part of it. Good question. There's one more big takeaway. What do you guys think that would be? Bold. So, fair question. So, glucose 4, there's a lot of different transporters, and we talked about them. For the purposes of this lecture, glucose 4 is very important because it is the transporter for fat and muscle tissues, and it is regulated by insulin. So when insulin levels are high, glucose 4 transporters are going to insert into the cell membrane, causing glucose to enter the cell. And then what's going to trap glucose in the cell? Phosphorylation of that 6th carbon, right? Any questions? Yes.
So, if glucose is high, is it just upregulation?
Say that again a lot louder. I can't hear you.
When glucose is high, is it upregulation of the GLBT4 protein?
Yeah, that's a great question. So what happens when there's a lot of glucose in the blood? Insulin is going to increase. That's going to cause the blood transporters to insert in the cell membrane to draw the glucose into the cell. And GLUT4 transports glucose in what tissue? The muscle and fat tissue, right? See, we're already done with glycolysis. Wasn't that easy? Okay, so there are some other, we talked about some other monosaccharides, fructose and galactose. Those can also enter the glycolytic pathway. So, and they do that, they are converted to intermediates of the glycolytic pathway. So fructose is converted to DHAP, dihydroxyacetone phosphate, and glyceraldehyde, and galactose is converted to glucose 1-phosphate. And so in muscle cells and in the kidneys, fructose is phosphorylated to fructose 6-phosphate, and that will enter glycolysis in the 3rd step, as shown here, fructose 6-phosphate, right? And galactose will interact with UDP to yield galactose 1-phosphate, then that will be converted to glucose 6-phosphate, and that will enter glycolysis in the 2nd step. Does that make sense? And here's just another representation of that. So we have glycolysis in pink on your right-hand side. Galactose will be converted to first glucose 1-phosphate, then glucose 6-phosphate, tetraglycolysis. Then in the muscles, fructose will be converted to fructose 6-phosphate. And in the liver, fructose will be converted to fructose 1-phosphate. Then it would be converted into glyceraldehyde or DHAP, and it'll be two of them, right? Because that's when it's broken down by aldolase into 2 3-carbon sugars. Any questions about that? All right. Here's the specific pathways. For fructose in the cytosol of the liver will be converted to fructose 1-phosphate by fructokinase. Aldolase B will convert that into the 2 3-carbon compounds, glyceraldehyde and DHAP, and those will be converted to glyceraldehyde 3-phosphate, which will enter the glycolytic pathway. And this occurs in the cytosol of the liver. So fructose is converted to glyceraldehyde 3-phosphate in the cytosol of the liver. Galactose is converted to first glucose 1-phosphate and then glucose 6-phosphate, which will enter glycolysis. Also, galactose can be converted to glucose 1-phosphate, which we will talk about later on in this lecture, how that can lead us to glycogenesis, which is the synthesis of glycogen. If there are any issues with this enzyme, galactokinase 1-phosphate uridyl we will have a buildup of galactitol, which is basically it's a product of improper metabolism of galactose, and that can cause cataracts. That's not the take-home message of this slide. Take-home message is galactose will be converted to glucose 1-phosphate and then glucose 6-phosphate and into glycolysis. Any questions? So when there are deficiencies in glycolytic enzymes, you have what's called glycolytic disorders. We have the phosphofructokinase deficiency, PFK deficiency, and pyruvate kinase deficiency, PK. So what happens in phosphofructokinase deficiency, there is the enzyme phosphofructokinase is defective, and so that's going to cause our muscles to fail because glycolysis cannot occur and produce ATP. That's going to cause muscle weakening. Then it's going to cause myoglobin to be released from our muscle cells. That's going to filter in the urine, and it's going to cause myoglobinuria, which is the dark color of the urine. This condition is typically seen after extensive workout, and it doesn't appear until like the teenage years and up to the 30s. Yes.
So is this wrapped up?
Yeah. Good question. Rhabdomyosin. And pyruvate kinase deficiency primarily affects the red blood cells because the red blood cells rely on glycolysis in order to produce ATP. And this last step, the pyruvate kinase step in glycolysis, is the ATP producing step in glycolysis. So red blood cells do not have mitochondria, so they cannot produce ATP through the electron transfer chain. So they rely only on the glycolytic pathway. And so what happens is because ATP can't be produced, that causes the red blood cells to lyse also happens is because oxygen is unable to be transported like it needs to be, there's an acid, the BPG or bisphosphoglyceric acid, that will increase in pyruvate kinase deficiency. And what this acid does is it will decrease oxygen's affinity to hemoglobin. So what that does is that will allow oxygen to be released in the tissue. And that's kind of counteracting the hypoxic nature that this condition causes because red blood cells are lysing, they're dying, and so there's not enough of them to move oxygen to the needed tissues. And so that's going to cause our red blood cells to break down. They're breaking down. And in babies, that's going to lead to jaundice because heme is being broken down. It's being converted to bilirubin and biliverdin, and that is not filtered from the body fast enough, so that's going to cause jaundice in babies. And because we're not getting enough oxygen and we're not able to produce energy, that's going to cause shortness of breath and muscle fatigue as well. Any questions? Yes.
So is PK typically seen in infants then, whereas PK is mostly in adolescents or?
That's a good question. I'm not sure if it's only seen in infants, but it would definitely be seen in infants because of the jaundice. It probably manifests as jaundice. But I think it might be seen in all ages. But that's a good question. I'll look into that. All right. Any questions so far? Look, we're almost halfway done with this pathway. That makes me want to dance. Y'all don't want to see me dance. Nobody wants to see me dance. Because I do old dances that were discovered before you were born. And I have a particular dance that my family hates because that's the only one that I do. I won't do that to you guys. I won't embarrass myself. Maybe later. So, say again. Okay, yeah, I'll do it. I'll do it at the end. But don't laugh at me when you see me. So now we're moving to another means of glucose metabolism. This is the pentose phosphate pathway. One of your classmates alluded to this earlier, pentose phosphate pathway. This pathway is also called the HMP hexose monophosphate shunt. And what this pathway does is that it produces NADPH. NADPH is an important antioxidant. And it also produces substrates that lead to ribose 5-phosphate production. And we know ribose is the sugar involved in nucleotides, right? Remember that from the last slide, right? Yeah. Any questions about the take-home message from the PPP pathway? So this pathway, pentose phosphate pathway or hexomonophosphate shunt, it occurs in the liver, mammary glands, and also the adrenal cortex to a lesser degree. It produces nucleotides, as I mentioned. It's also involved in fatty acid synthesis and breakdown and cholesterol synthesis. And that NADPH is a very important antioxidant. We're going to talk a lot about NADPH in block 3, but just know it's very important as an antioxidant. It donates its hydrogens to glutathione. Glutathione is the actual antioxidant that our body needs to get rid of free radicals. So, this pathway, glycolysis is here, and this is the pentose phosphate pathway or the hexomonophosphate shunt. What happens is glycolysis. In glycolysis, we know glucose is converted to glucose 6-phosphate. The enzyme glucose 6-phosphate dehydrogenase, or G6PD, will convert glucose 6-phosphate to 6-phosphoglucanolactone. When it does that, when this reaction occurs, it produces NADPH, which is very important, as I mentioned, for cholesterol biosynthesis, fatty acid biosynthesis, and it serves a role in being an antioxidant. And then this pathway, this intermediate is converted to ribose 5-phosphate, which is used to produce nucleotides for DNA and RNA. Any questions? So somebody, yes.
Yeah, when does G6PD? When does it actually pulling glucose 6-phosphate out of glycolysis? Is it like a negative, there's too much of it, like we don't need glycolysis, so we'll start shunting it over?
Perfect question and answer. You answered it. Yep. So when there's enough glycolysis being broken down to produce energy, this pathway is going to shunt into the pentose phosphate pathway to produce that NADPH and produce the reagents for nucleotide synthesis. So anybody loudly tell me what the take-home message is for the pentose phosphate pathway. Say again. Biosynthesis of what? Okay. What else? Why are you laughing? Well, you're correct. No, you're definitely correct. There's one more thing. No, everything you said was correct. There's one more thing that is important. Produces NADPH. I tried to bold it, but I don't know if you can. Is it bold? It is, okay. Yeah, so, yeah. Yes.
I'm having difficulty understanding, I guess, like how ribose 5-phosphate correlates to fructose 6. Yeah, that. Why is that connected by the two arrows? Like, is that ribose like synthesize those two things on the left side?
So ribose 5-phosphate can be converted back to glyceraldehyde 3-phosphate and fructose 6-phosphate, which you mentioned so beautifully. That's not the important part. It is important, but not for our purposes, but it can occur. Now, I can go over those steps if you guys want me to. It's not going to be on the test.
Absolutely not.
You sure? So just know that that ribose 5-phosphate can be converted back to intermediates of glycolysis. That's correct. So you don't need to know the details. You don't even really need to know that in terms of what we're talking about. But yes, that can occur. Any questions? So, does anybody have a friend, family member with G6PD deficiency? Yes. So, if you don't mind sharing, are there any symptoms or characteristics that you notice with them in particular? You are. You stole the thunder. Thank you for saying that. That's great. Absolutely great. So, G6PD is commonly found in Black African American men. Some people have symptoms, but as your classmate mentioned, some people don't. I didn't actually know that some people don't have symptoms since I've been here doing research about it, because I got asked the question. And I didn't know that there's like 6 different categories of enzyme activity, and some people have no symptoms, some people don't even know until they actually get the test done. This is one of the tests that we were supposed to get done when we joined the Army. Some people get it done every time they're deployed, but this doesn't change. If you're G6PD deficient, then you're going to be G6PD deficient for the duration of your life. And so when I was in the Navy like 30 years ago, this is the first time I heard about this. And this was in boot camp, and one of my shipmates in boot camp, he had it, and he had exercise intolerance. So he was always getting in trouble. I mean, always. And so I remember one time, I don't know what he did, but I remember it like it was yesterday, because got in trouble, and the drill sergeant made him exercise, and we were all in our berthing, that's what we call the bay in boot camp. And he's exercising, doing jumping jacks in front of us, and like he kept acting like he was gonna pass out. Or not acting like but he kept stopping. And so we were getting worried. I was kind of worried about him. And the drill instructor was like, that's okay, let him pass out. That's why we have ambulances. And that just stuck with me, because he didn't pass out, but he got in trouble like all the time, and he always had to do exercise. Like he never learned that if you have exercise intolerance, you're gonna want to follow the rules even more. Nobody wants to be doing push-ups right? Nobody wants to be doing push-ups until you're fatigued and you can't lift your arms, and he always got in trouble. And so people used to make fun of him and like yell his name, so the drill instructors would be in another room, they would hear, and they would come in there and be like, Jenkins, what are you doing? And he would be doing nothing, but they were trying to get him to be doing that. Jumping jacks, he be doing, he's like, he do it again. and the drill sergeant was like, the drill petty officer was like, you've done 2 jumping jacks, you're gonna be fine, and if you pass out, that's okay. That just stuck with me, because I'm like, wow. And after 7 weeks of him doing that, I'm like, yeah, he wants to get in trouble. You wouldn't keep doing that. But anyway, that was a very interesting story. My first experience with G6PD. I don't even know if he had exercise intolerance or not, because he liked to do it clearly, but he pretended like he did, maybe. But no, it's not making light of it. But that is one symptom of those that actually have clinical manifestations of G6PD. They do have exercise intolerance. And they also have red blood cell lysis, which of course will cause jaundice in a baby, cause fatigue. We know that there's going to be a decrease in that antioxidant agent, NADPH, right? Because the reaction can't go this way because this enzyme is deficient, right? That makes sense. And then there's going to be Heinz bodies and bite cells. Basically, Heinz bodies shown here are degraded hemoglobin. And so what happens is that those little purple, dark purple spots are the degraded hemoglobin. And the bite cells occur when the spleen macrophage molecules will bite out that degraded hemoglobin. So you see it actually looks like pieces were bitten out of the red blood cells, and that's the macrophages from the spleen biting out those degraded hemoglobin cells. Any question about G6PD deficiency? Yes?
I had a teammate with this. He had 0 symptoms, but I wasn't allowed to give him his Zeds.
Why not? That's the last bullet. Perfect. Why not? You know why not? Because NSAIDs will cause, thank you for that segment, that was perfect. NSAIDs, we're going to talk about this in the next slide too, but NSAIDs are oxidizing agents, they can be, and so what that means is that they can potentially cause free radical buildup in the body, and therapeutic doses of NSAIDs won't do that, but because these people that have G6PD deficiency already can't produce enough of the scavenger or the antioxidant, you're not going to want to give them anything that can potentially cause free radicals. Does that make sense? All right, and also because of the hemolysis of the red blood cells, there's also going to be dark urine present in these sufferers as well. Yes.
Is this like a spontaneous disorder, or is this something that's like genetic?
It's X-linked recessive disorder. So it can occur. Like I also during my research, I thought it only occurred in Black African Americans, but it can occur in anybody. So it can. I don't know if spontaneous is the correct word for when it's found in other ethnicities or races, but it's definitely something that's rare. It's rare, but it's also the most common enzyme deficiency that causes anemia. So that's kind of weird. But yes, it's something that that is X-linked, and it will affect. It can affect the ability of a person to exercise. Does that answer the question?
Yes, sir.
Any other questions? Yes.
Sir, is this also a contraindication for tetracycline antibiotics also?
Absolutely, for the same reason. So they're gonna cause potential oxidation or oxidizing, you know, and so there's not gonna be any scavengers to get rid of those oxidizing agents. So you're not gonna want to give them sulfonamides and drugs like that. Any other questions? All right, you guys take a 10-minute break.
Literally, the question is,
Right. And you have to
you're still teaching. Because none take the confidence. Yeah, that's just me always talking. You ask him, it's like, dude, if you're really that interested, you have a safe. Have your right, the part goes in freaking way far. And the part all the way there. Gonna go to the grocery store after class.
I mean,
Getting low on my lunchbox though, so I didn't pack a lunchbox because I didn't really want to.
I think it's more of a that I could be wrong. They might be there, but just know that they're gonna be like inserting that they're gonna cause you, because that's typically these glucose transport. And we're gonna talk about this in the last one. If they from my that's yeah. Yeah, no one. Wait a minute, what was that?
I used to see Steve if we had the opportunity to take the bus, but I had romantic days here. And they gave us.
When are we going to the field? When are we going to Mike does that. See what I said? Who's fava beans? Huh? Who's fava beans? It's like the way that the blood flows out blood. Oh no! Fava beans are like actual beans that they eat. What is it? It's just a type of bean. They're like green.
Last one is the
At the moment? Kind of.
oh, dude, someone prepared. Oh, just I was watching last week. It's okay.
Random ass names that'll make it worse.
I was like, dude, yeah. So I asked him,
All right, guys. Let's get back into it. Any questions about G6PD deficiency? No? Want me to do a cartwheel? Oh, not a cartwheel. Jumping jacks. It's too hot in this room. It's hot. No questions? So, pyruvate is the final product of glycolysis metabolism. How many pyruvate molecules do we gain per one glucose molecule? 2. Correct. And so, if oxygen is present, that pyruvate is going to be converted into acetyl-CoA, as Lieutenant Bishop mentioned. When oxygen is not present or when it's low, that pyruvate will be converted into lactate. We're going to talk about what happens under aerobic conditions now, and at the end of this lecture, we're going to talk about the Cori cycle, which is the pathway where pyruvate is converted to lactate. So here is the reaction here. You have that pyruvate molecule, which is the final product of glycolysis. It moves from the cytosol to the mitochondria, as O.C. Mitchell mentioned, and it interacts with that thiol group from the CoA, producing acetyl-CoA, and in this reaction, catalyzed by pyruvate dehydrogenase, NAD plus is reduced to NADH. And so, when oxygen is not present or when we're at low levels, we're struggling to exercise and take the, it's not, I guess, AFT now is what it's called as of June, right? AFT instead of ACFT. Y'all don't have any struggles with this, but I do because I'm old and slow. So I have a lot of oxygen deficit. My muscles are sore before I even finish the test, which is a bad sign. That just means I'm out of shape too. So when oxygen is low, that pyruvate is reduced to lactate. The reaction is here. Pyruvate, in the presence of the enzyme lactate dehydrogenase, produces lactate. And this reaction uses NAD. So because we know NAD, as I mentioned earlier, is important for energy production, basically we lose one molecule of NAD if oxygen is not present, and we're going to talk about that in the last lecture of this block when we talk about the electron transport chain. So basically, under aerobic conditions, pyruvate is converted to acetyl-CoA. Under anaerobic conditions, pyruvate is converted to lactate. Okay, and so under anaerobic conditions, this lactate will basically, it's going to take away one of our NADH molecules, and so that's going to decrease the amount of ATP our body can produce. And again, we're going to talk about that in the last lecture of this block. So now we're going to talk about glycogenesis and glycogenolysis. So we already have three words that sound the same: glycolysis, glycogenesis, glycogenolysis, and there's going to be one more. So what's glycolysis? The breakdown of glucose. So glycogenesis is the synthesis of glycogen for later use. Insulin regulates glycogen synthesis, so when we have a lot of blood glucose, insulin will be released. That's going to cause our cells to take in the glucose, and it will be stored in the form of glycogen in the liver. So the liver cells are going to make glycogen. Glycogenolysis is the breakdown of glycogen into glucose 1-phosphate. And this is activated by glucagon, which is another hormone, and epinephrine. So during fight or flight, that epinephrine will be released, and that's going to cause our body to break down glycogen so we can get out of dodge or get out of whatever we need to do, fight or flee. That's how that works through epinephrine and glucagon release. Any questions about that? So we talked about glycogen. It's a heavily branched glucose polymer with alpha 1,4 and alpha 1,6 bonds. How frequent are the branches? Does anybody remember? Hey, look and see, y'all do remember. Look at that. Y'all acting like y'all didn't. Good. Yep, 10 to 15. So glycogen is formed when there are high levels of glucose 6-phosphate. And that means that our body has enough glucose. And so when we have enough glycogen in our body, any excess glucose is not going to form more glycogen. It's going to make you look like me. Right? That glucose is going to be converted to fat. Right? Right? That's what it says. So, if there's enough glycogen, then our body is going to convert that additional glucose into triacylglycerols or triglycerides, and it will be stored as fat. Any questions about that? Again, Glycogenesis, synthesis of glycogen for later use. Glycogenolysis is the breakdown of glycogen into glucose, and that occurs in our liver. And here is a little schematic of the reaction. So we have glycolysis, that glucose 6-phosphate. What's important about glucose 6-phosphate? One thing that we talked about. It traps glucose into the cell, right? So when we have glucose 6-phosphate, if we have a lot of glucose 6-phosphate, it will be converted to glucose 1-phosphate, and then UDP glucose in the presence of glycogen synthase and high levels of insulin will make glycogen. All right? Now, if we have low glucose levels, low blood glucose levels, or if we need to fight or flee, which means that epinephrine levels are high, or glucagon levels are high, or cyclic AMP levels are high, those are all stimulators of glycogenolysis. Those are going to cause glycogen to break down to glucose 1-phosphate by glycogen phosphorylase. That glucose 1-phosphate will then be converted to glucose 6-phosphate in order to continue down the glycolytic pathway and produce energy in the form of ATP. Any questions about that? So what's the enzyme for glycogenolysis?
Phosphorylase.
Anybody?
Glycogen phosphorylase.
An enzyme for glycogenesis? Okay. Any questions? All right. Check on learning. So match each description with the following terms. What is activated by low levels of blood glucose? Say, one person say it. Glycogenolysis. Yep. What about converts glucose 1-phosphate to glucose 6-phosphate? Glycogenolysis. What is activated by high levels of glucose 6-phosphate? Glycogenesis. And what converts glucose and UDP to UDP glucose? What pathway is that involved in? Glycogenesis. Any questions about that? Does that make sense? All right, so here are a number of glycogen storage diseases. I recommend that you guys look at this slide, know the differences between the defective enzymes, what the disease is, and the symptoms. Pay close attention because some of them, the same enzymes are affected, but it's in different tissues. So, for example, Ricardo's disease cannot degrade glycogen for use in muscle cells. So there's going to be a clinical manifestation of exercise-induced muscle cramps and exercise intolerance. Yes?
So what is the type in reference to?
Somebody wanted to give it an extra name. So sometimes they're identified by type 0, 1, 2, 3, or they're just called by the defective enzyme name. So it's just whoever, I don't know if it was different people naming them in different places in the world. And so one of them named them by Roman numerals, the other one named them by defective enzymes. So that basically type 5 or type 6 would be the glycogen phosphorylase in the liver. So.
Do you ever hear them referenced by type, or is it more so that, what do you, I know you're not a clinician, but what do you typically hear?
I typically hear them by the defective enzyme, to be honest with you. And then they would have to say muscle, liver, So, but I think, if I'm not mistaken, depending on where you are clinically, they can be named or stated using the type or the enzyme reaction. Any questions about this table? Gluconeogenesis. So this is the fourth glucoglyco word that added confusion to me when I was in grad school because they all sound similar, especially when you're about to take the exam. They all look the same. So gluconeogenesis, this is synthesis of glucose from a new source. That's how it got its name, gluconeogenesis, and that is when glucose is synthesized from a non-carbohydrate source. For example, could be lactate, could be pyruvate, could be glycerol, and or even amino acids. So I underlined amino acids and glycerol. Why do you think I did that? Just like, what's the importance of these two?
Tone test.
Proteins and fats. So this is how the macronutrients are converging together to create glucose to be used to make energy. Does that make sense? Okay. So this reaction occurs primarily in the liver, and this happens when all of the glycogen stores are depleted, and so basically your body needs a new source of glucose in order to obtain energy, and so it will use gluconeogenesis, and this reaction is not the reverse of glycolysis. Why not? Why wouldn't it be the reverse of glycolysis? One person? Because there are irreversible steps in glycolysis. That makes sense, right? So, gluconeogenesis, there are 3 irreversible steps of glycolysis that gluconeogenesis needs to circumvent in order to produce glucose. And so, remember the steps that are irreversible in glycolysis are steps 1, 3, and 10. So, the first step of gluconeogenesis is circumventing step 10 of glycolysis, and how it does it through the enzymes pyruvate carboxylase and phosphoenolpyruvate carboxykinase. And what happens is pyruvate will be converted to oxaloacetate. Remember oxaloacetate, remember that name, we're going to see that again in future lecture. And I'm going to ask you where you saw this, so make sure you remember this. Also, oxaloacetate will then be converted to phosphoenolpyruvate, and the second step of gluconeogenesis to produce by the enzyme phosphoenolpyruvate carboxykinase, as you can see here. So it's just circumventing this last irreversible step of glycolysis. And so you see all the other steps in between are irreversible. So now step 2 of gluconeogenesis needs to circumvent step 3 of glycolysis, which was that irreversible step. So what that is going to do, that's going to occur through the enzyme fructose 1,6-bisphosphatase, and that's going to convert fructose 1,6-bisphosphate into fructose 6-phosphate, and then the final step, glucose 6-phosphatase is the enzyme in gluconeogenesis that will convert glucose 6-phosphate into glucose. And so here's the reaction for that first step. Pyruvate is converted to oxaloacetate by pyruvate carboxykinase. Oxaloacetate is converted to PIP by PIP carboxy kinase. And here is the glycolytic pathway, and the gluconeogenic pathway. Glycolysis is going down, gluconeogenesis is going up, circumventing the 3 irreversible steps. The first step it does the gluconeogenesis circumvents it using 2 enzymes, so it's a 2-step process. And then the enzymes are pyruvate carboxylase, PIP carboxykinase, and then fructose 1,6-bisphosphatase, and then glucose 6-bisphosphatase. Any questions on how the gluconeogenic reaction occurs? Yes.
So this is talking about turning pyruvate back into glucose. We're talking about the other half of the genetics. Is that breaking it down into the
It can, or it can break them down to other intermediates, and we're going to talk about, that's a great question, we're going to talk about that in a later lecture, but it can either kind of convert them to pyruvate, the ones that can be converted to pyruvate, or it can convert them to another intermediate that will eventually get us to glucose. We're going to talk about that later. Any other questions? Yes.
Sir, can you go explain again how this is not the opposite of glycolysis? So this one is creating glucose, the other one is breaking down glucose. How is it not the opposite of it again?
Because it's using different enzymes in the irreversible steps of glycolysis. That's all. So it's there. You see, there's a lot of steps that are the same, where they can go back and forth, but there's 3 steps that are irreversible in glycolysis. So gluconeogenesis needs to find a way to circumvent those steps, and it's doing that through these 2 enzymes here, this enzyme, and then glucose 6-phosphatase. Does that make sense? Any other questions?
Does the whole process use ATP, or is it just specific steps? Or does it not matter?
Just the specific steps. So that first step uses ATP and GTP, which is ATP. So any other questions? All right, here are the differences between glycolysis and gluconeogenesis in terms of how they're regulated. You see gluconeogenesis, the important enzymes are the ones that just mentioned, glucose 6-phosphatase, fructose 1-bisphosphatase, and then pyruvate carboxylase. Those are all regulated by low blood glucose levels. Remember, gluconeogenesis will occur when we don't have enough glucose in the blood. And then glycolysis, the enzymes of importance are hexokinase, phosphofructokinase, and pyruvate kinase. We know that hexokinase is regulated by insulin, or activated by insulin and high glucose levels. It's inhibited by high glucose 6-phosphate levels. PFK is activated by AMP and ADP and inhibited by ATP, and pyruvate kinase, or PK, is activated by fructose 1,6-bisphosphate and inhibited by ATPs. And then finally, the Cori cycle is a cycle that converts that lactase that was created during anaerobic conditions in our muscles. What happens is this lactate in the muscle is brought to the liver, converted to pyruvate, then converted back to glucose. That glucose can then be stored as glycogen if need be, or it will travel back to the muscle to provide oxygen and glucose to the muscle. Any questions about that? All right. Check on learning. So identify each of the processes below. What is the synthesis of glucose from non-carbohydrate? Gluconeogenesis. What about the breakdown of glycogen into glucose? Glycogenolysis. Oxidation of glucose into 2-pyruvate. Glycolysis. And the synthesis of glycogen from glucose. Glycogenesis. Do you guys have any questions? Yes.
So this might be a little off topic, but I knew a guy that his body didn't produce lactic acid. So is that something in the Krebs cycle where his body just didn't have the lactic dehydrogenase enzyme?
Probably not. It'd have to be something like that. That's a good question. I'm gonna look that up. I've never heard of that. So he never got tired?
Yeah, we'd do like PT tests and stuff, and he would do like 90 push-ups, and be like, yeah, I feel fine. But he never got like, his body seemed to never like get weak.
It's a robot.
It
I'm gonna
causes
look that
problems.
up.
He said he was like,
He said what?
yeah, I asked him about it one day. He said, he got like tested, and they said like his body doesn't produce lactic acid, so his muscles didn't get tired.
So he maxed out with everything.
Yeah, 100%.
I never did that. Never will. Even when I was you guys. That's a thank you for that. I'm gonna look into that. That's very interesting. I've never heard of it. Any other questions, comments? All right, I'll see you guys tomorrow.